topic we are going to cover is human health and disease from 12th Zoology. And before entering into today's chapter, so yesterday someone asked about Balbiani rings. Okay, so where these are present means in polythene chromosomes. So what are polythene chromosomes? So these polythene chromosomes, they have multiple copies of chromosomes. Multiple copies of chromosomes. So how it happens means, that is, they say in interphase. Cell division does not take place, but synthetic phase produces multiple copies of DNA. So that leads to the division does not take place. So inside the cell, multiple copies of chromosomes will be there. Okay, 10 to 20 times the division will take place, and enlargement of cell will take place. So these are the Balbiani rings which are responsible for RNA synthesis. Okay, which are present in these polythene chromosomes, where these are present in salivary glands of insects. Salivary glands of insects. Is it clear? Okay, so let us move on to the today's topic, human health and disease. An autoimmune disease is Myasthenia gravis, Hemophilia, AIDS, none of these. So first, what do you mean by autoimmune disease? So generally, antibodies are produced against antigens. Against antigens. So what do you mean by antigens? Generally, foreign particles which are produced by any bacteria or viruses are called as antigens. And there are some antigens which are present in our cells also. They are called as self-antigens. They are called as self-antigens. So generally, these antibodies should be produced to remove the infections. Okay, infectious organisms or infections. Okay, so here in autoimmune disorders, these antibodies are produced against the self-antigens. So automatically, what will happen if antibody and antigen meet means that will lead to agglutination. Lead to agglutination. It is a kind of precipitation. Okay, so this is called as autoimmune disease. So 3 diseases are given. AIDS, you very well know that it is caused by the virus, human immunodeficiency virus. Okay, it is not an autoimmune disease. And what is hemophilia? So hemophilia is otherwise called as royal disease or factor VIII deficiency. So that means you know very well that clotting mechanism requires so many factors. So in that, one of the factor is deficient, so that the clotting will be blocked. Because of that, hemorrhage will happen if any cut is happening on the body. Okay, that is called as hemophilia. So why it is called as royal disease? It is there in the family of Queen Victoria, and generally why it is happening? Due to the genetic disorder. So the gene which is producing this factor is having some genetic disorder, genetic defect, like mutated. Okay, so this is a genetic disorder. It is not autoimmune disorder. Actually, X-linked recessive. So here, Myasthenia gravis is the correct answer for autoimmune disease. What is Myasthenia gravis? So Myasthenia gravis, if you take impulse transmission, you know the mechanism of impulse transmission. So generally the presynaptic neuron will release acetylcholine, clear. And here in neuromuscular junction means neurons, that is nervous system, connected to the muscle cells. Okay, so that is to carry out the work, we have to receive the signals from our nerves. Our muscles should get the signals from nerves. So every muscles are having connection with neurons. These neurons releases acetylcholine, and the acetylcholine receptors are present on the muscle cells. This acetylcholine should bind to the receptor to carry out the function. But here in Myasthenia gravis, what is happening means, so this is the receptor which is present on the muscle cell. Okay. And actually, you know that receptors are proteins. Antibodies are produced against these receptors. So for example, this is the antibody produced against this receptor. So this will come and bind. Actually, when the receptors are bind with the ligand, the cell, muscle cell gets activated. Okay, so here it should be activated only when it is needed. When it is needed, acetylcholine will be produced from neurons. But here, due to the production of antibodies, always it is excited and it gets weakened. That is called as myasthenia gravis. Okay, which of the following immunoglobulins is present in milk? So generally it is given as milk. It is mother's milk. Okay. So what do you mean by immunoglobulins? So you know very well that blood is made up of plasma and formed elements Okay. So if you take the composition of plasma, 90 to 92% of water and remaining are of proteins, amino acids, lipids, carbohydrates, etc. Okay. So in these proteins, if you take, there are different types of proteins present in blood. One is fibrinogen. What is the role of this fibrinogen? It is involved in clotting mechanism. And the second one is albumin. Okay. So what are the major role of albumin? Maintaining osmotic pressure and thereby osmosis and blood pressure is maintained by albumins. And the third type is globulins. So these are involved in defense mechanism. Okay, so that is immune function. The proteins which are involved in immune function are called as immunoglobulins. There are different types of immunoglobulins: IgA, IgG, IgM, IgE, IgD. Okay, so why they are differentiated like this means in their structure they have light and heavy chains. So the nature of amino acids which are present in the heavy chain varies in the different immunoglobulins. So here the immunoglobulin present in the mother's milk is IgA. So due to this IgA, infants are getting immunity. Okay, and actually there are different types of immunity. Let us see later in another question. Okay, so here the babies are getting immunity directly from mother. That is called as natural acquired immunity. Acquired means receiving. So naturally the babies are receiving immunity from mother. So this type of immunity is called as naturally acquired immunity. Clear? And the third question is, most bacteria ingested with food are killed by. Most bacteria ingested with food are killed by, because so many openings we are having. So each and every part is having some mechanisms to protect against bacteria or viruses, any microorganisms which are causing diseases to us. Okay. So here, what are the options given? Cilia and mucus on the lining of the tract. So what do you mean by mucus? Mucus that is rich in mucin protein. This helps to prevent the damage caused by HCl. This you might have learned in digestive system. Clear? And stomach acids. What is the major acid produced by stomach? HCl. In gastric juice, HCl is there. So due to this, the pH of the stomach is very low. Automatically, the organisms will die. The bacteria cannot withstand the pH produced by the produced by the stomach, that is gastric juice. The intrinsic factor in the stomach. So what do you mean by intrinsic factor? Again, intrinsic factors are produced by oxyntic cells or parietal cells of the stomach. Okay, what is the role of this intrinsic factor? This helps in the absorption of B12, which are produced by bacteria in our gut. Okay, so what is the role of this B12? That is very much needed for the production of RBC. So if the B12, vitamin B12 is not absorbed properly, automatically that will lead to anemia. Okay, so bile in the small intestine. So from where we are getting this bile? Bile is secreted by liver and stored in gallbladder. Clear? And what is the major role of bile? Involved in the digestion of lipids. Digestion of lipids. Okay, so again, the question given here is most bacteria ingested with food are killed by stomach acids. And even I can explain, like there are different mechanisms which will protect us from any infections. And again, in immunity, we are having two different types of immunity, that is innate and acquired. Innate and acquired immunity. So what do you mean? What do you mean by innate? Innate means by birth we are having some immune system. So what are they? They are like physical barriers, physiological barriers, cellular barriers, and cytokine barriers. Okay, so what do you mean by physical barriers? The entire body is covered with skin. So what is the major role of skin? It prevents the internal system. Okay, so the skin is the physical barrier. Physiological barrier means the secretions, for example, tears which are produced from tear gland. It is rich in lysozyme. Lysozyme is the enzyme which is present in lysozyme is the enzyme which is present in tears, saliva. What is the role of lysozyme? It kills bacteria. Clear? So you have to know about the different kinds of barriers. So stomach acids, physiological barrier, for example, saliva you can say, tears you can say, stomach acids you can say, even vaginal secretions you can say. Okay, cellular barriers, again T cells and B cells. What are cytokines? Cytokines are the secretions which are protecting us against infections. Okay, so the most bacteria ingested with food are killed by stomach acids. The letter T in T lymphocyte refers to thalamus, tonsils, thymus, thyroid. So here you have to know about organs, lymphoid organs. Lymphoid organs. So what are lymphoid organs? The organs which are produced, which are involved in immune system. There are two different types of lymphoid organs, primary organs, that is thymus and bone marrow. Thymus and bone marrow, because the immune cells are produced from these two organs. Okay, and the secondary lymphoid organs are helping us to store the cells which are going to protect us. Okay, so what are they? They are tonsils, lymph nodes, okay, spleen you can say, and Peyer's patches in intestine. Okay, so the letter T, lymphocyte, T in T lymphocyte. What do you mean by T cells and B cells? So B cells, they are involved in humoral immunity. Humoral immunity. What do you mean by humoral immunity? Humoral immunity means they are producing antibodies into blood. These antibodies will fight against antigens and protect us. Okay, and second cells which are involved in immune system is T cells. Okay, so what do you mean by T cells? Again, there are different types of T cells, T helper cells and cytotoxic T cells. Okay, these are produced from bone marrow. Okay, and B cells are also produced from bone marrow. These are produced from bone marrow, and they are matured and differentiated into cytotoxic or helper T cells in thymus. So where this thymus is present? Near the sternum. So this thymus, it is bigger in size in children. In children, it is big in size. And if you take old age people, the size will be smaller. Okay. And this is the right answer actually. What do you mean by tonsils? So where this tonsil is present? It is present on either side of the pharynx, wall of the pharynx. Okay. The major role of tonsil is, again, it is also involved in protection. Okay. They releases, they are having cells to protect against the infectious organisms. So what do you mean by thalamus? Thalamus is the part which is present in central nervous system. It is having many nuclei which controls many activities of the body. And thyroid, this is the gland, you know very well that it secretes T3 and T4. These hormones regulates many metabolic reactions. Okay. So here the letter T in T lymphocyte refers to thymus because it is matured. T lymphocytes are matured in thymus. Which of the following is a pair of viral diseases? So in this chapter, you have learned about viral, bacterial, fungal, protozoan diseases. Okay. So here different combinations of diseases are given. The first disease is common cold and AIDS. So generally common cold is caused by even you can say coronavirus. Okay. And AIDS is caused by human immunodeficiency virus. Okay. So the answer, correct answer is common cold and AIDS. Let us see about the other combinations. In B, dysentery. So dysentery is caused by Shigella. Okay. So Shigella is a bacterium. Again, common cold is caused by virus. Okay. Typhoid. So typhoid is caused by Salmonella typhimurium. This is also a bacteria. Okay. What do you mean by tuberculosis? And again, Clostridium tuberculosis. And what do you mean by ringworm? So again, yes, the ringworm is caused by fungi, such as different fungal organisms causes this ringworm. Okay. They are Microsporum and Trichophyton, Epidermophyton. Okay. So the correct answer is common cold and AIDS. Artificial immunity can be acquired from a serious illness, vaccination, repeated exposure to the same microbe, treatment with penicillin. So penicillin is antibiotic. Antibiotic which can easily kill the infectious organism. Okay. So before entering into these options, you have to know what kind of immunities are present. Okay. And again, in artificial immunity also, you should know about, that is innate immunity. I explained already in a question. Let us see about acquired immunity. Okay. So here acquired immunity means during our lifespan, we are getting immunity. When we are getting infections, automatically our body is fighting against the infections and the immunity is developed. That is called as acquired immunity. So again, this acquired immunity is of two types. That is, it occurs naturally or it occurs artificially. And also it is of two types, passive and active. So what do you mean by passive? Passive means automatically, directly we are giving antibodies to the patient so that the body is not going to work to produce antibodies. Directly we are helping the body to kill the infectious organisms. That is called as passive. Okay. And this is also of natural and artificial. Natural and artificial. So what do you mean by natural and artificial passive immunity? So natural means automatically my body will, for example, children, babies, infants, exactly I can say infants. So they are getting naturally antibodies from mother's milk. This is called as natural passive acquired immunity. So what do you mean by artificial passive acquired immunity? So if I'm getting infected to some disease, I can get preformed antibodies. So now preformed antibodies are available. For example, you can say tetanus. and snake bite. If I have any snake bite, so preformed antibodies are there. So snake venom is an antigen. Okay, so after getting into the body, that will suppress my immune system, and it will create some other disorders. Okay, so that will be easily suppressed by the direct application of these antibodies, preformed antibodies. Okay, and what do you mean by active? Again, this is also two types, natural and artificial. So natural and artificial here, naturally, if I'm infected to typhoid or any other disease, automatically my body will work and produce antibodies against this infectious organism. Okay, so this is called as natural active acquired immunity. And what do you mean by artificial active acquired immunity? See, now we are getting vaccines for coronavirus. Even the persons, those who are not infected, we are getting vaccines. Okay, why? We are getting ready with our body to protect against the virus. If it is going to infect us, already we are having protection in our body. Okay, so directly there are different types of vaccines are available. Okay, so this is called as, if you get vaccine, automatically our body will produce antibodies against that organism. In future, if you are infected with that organism, the already produced antibodies will fight against that. This is called as artificial active acquired immunity. Okay, and this is the correct answer actually. Artificial immunity can be acquired from a vaccination. You have to know what is vaccination. There are different types of vaccines available. So what are they? How we are going to produce vaccination means by biotechnological techniques, live attenuated vaccines. So there are 4 different types of vaccines: inactivated or killed vaccines, even inactivated toxins we can inject, and even subunit or conjugate. So let me explain. What do you mean by live attenuated vaccines? So if a virus is there, we can remove a part which is responsible for producing disease. Okay, so this is called as attenuated virus. Okay, attenuated virus, which is going to act as vaccine. So if you are getting this attenuated vaccines, my body will produce, because this virus will be having so many proteins. This part which is having disease causing protein, if you are removing this protein, other proteins will be there. Automatically my body will produce antibodies against these antigens which are present on the virus. So when this virus enter in my body, into the body, automatically the antibodies produced already in my body will go and attach and destroy the virus. That is the mechanism involved in live attenuated virus. Inactivated or killed virus, so the virus will be killed and the pathogenicity will be removed and used as vaccine. Inactivated toxins. So if you see, most of the diseases are caused by the toxins produced, or antigens, you can say, produced by the organisms. Okay, so if you identify the toxins, you can remove the toxicity of this toxin and you can use it as vaccine. Okay, and again, subunit conjugate means exactly particular part. For example, so many vaccines are available for COVID. Okay, so if you take Covaxin. So Covaxin is actually attenuated vaccine. Okay, so killed organism, entire organism, they are introducing into the body. And if you take Covishield, that is a subunit or conjugate. That is a part of virus they are sending. Part of virus they are sending. So subunit means maybe toxins. If you take glycoproteins, toxins are mostly proteins. Glycoproteins, they are having different subunits. Maybe it has 2 subunit means. So one subunit they will derive and use as vaccine. Clear. So artificial immunity can be acquired from a vaccination. Which of the following organs is not involved in the generation of immune response? Okay, already I explained what are primary and secondary lymphoid organs. Okay, lymph nodes, spleen, and thymus. So these three are lymphoid organs. Thymus is primary, and spleen and lymph nodes, if you take, these are secondary lymphoid organs. So brain is not involved in immune response. If a person shows a production of interferons in his body, the chances are that he has got infection of thyroid, influenza, tetanus, malaria. Okay, so let us see one by one. What are interferons? So what are interferons? So these are the glycoproteins. Again, these are glycoproteins. So from where these glycoproteins are produced? Virus-infected cells, or even you can say cancer cells. Okay, so why these cells are producing these interferons? To protect the neighboring cells. Okay, so you know what is neoplastic transformation? So neoplastic transformation means normal cells, they are transformed to the malignant cells. Okay, that is, a clump of cells are produced. This transformation is caused by so many agents. This is called as neoplastic transformation. So this transformation can be prevented by this interferons. So generally, virus infected cells produces interferons to protect the cells. For example, this is virus infected cell. All other neighboring cells are having receptor for interferon. So this cell will produce, this is virus infected cell, this cell will produce interferon. This will go and bind to the receptor. That is the signal for other cells to protect. They will release toxins for, that is toxins for infectious organisms. Infectious organisms will be killed with the help of the processes induced by interferons. Okay, so here, which is caused by virus. Actually, I explained that interferons are produced only during viral infection. In these, influenza is caused by influenza virus. So again, Clostridium tetani is a bacterium. Thyroid, it is again T3 and T4 hormone producing gland. Okay, and it does not produce interferons. Okay, infections of thyroid. Thyroid is not infected. And again, tetanus, I explained. Malaria, it is caused by Plasmodium vivax, that is protozoans. Plasmodium vivax or falciparum. Okay. So the correct answer is influenza. Which of the following statements is correct? Benign tumors show the property of metastasis. Heroin accelerates body functions. Malignant tumors may exhibit metastasis. Patients who have undergone surgery are given cannabinoids to relieve pain. So let us see one by one. If you take cancer tumor, there are two types. One is benign and another one is malignant. What is the difference between benign and malignant? This is localized. Okay, this is localized and this can spread. That is the difference. Okay, so what do you mean by metastasis? So metastasis is nothing but spreading. Spreading. Okay, so for example, if one part of our body, for example, you can say my ovaries are having cancer. Okay, and this may be benign. If it is benign, the tumor will be present only in the ovaries and it is not spreaded to the nearby organs. But if it is malignant tumor, it is spreaded to the neighboring organs, like uterus will be affected, okay, and even the liver will be affected if it is not diagnosed in early stages. Clear? So benign tumors show the property of metastasis. It is wrong. Actually, malignant tumor shows the metastasis. So metastasis means even this ovary will produce the cancerous cells, and even these cancerous cells travel through the blood to some other part, okay, or else some other agents which will cause the normal cells to cancerous cells, they are released into the blood, and blood transports those substances to the neighboring part, and it is getting spreaded. That process is called as metastasis. Generally, the benign tumors are localized. Okay, so this is wrong. And heroin accelerates body function. Heroin. So if you take drug, heroin actually slows down the process, body functions. Malignant tumors may exhibit metastasis. Yes, correct. Patients who have undergone surgery are given with not cannabinoids. Morphine is used as painkiller. Morphine is used as painkiller. So next question, lysozyme kills by destroying. So I already explained that lysozyme is present in tears, saliva. Okay, so these are physiological barriers. Physiological barriers. Clear. So lysozyme kills by destroying. What is the major mechanism of lysozyme? If you take bacterial cell wall, is made up of, it is not having cellulose, it is made up of peptidoglycan. So if you take bacterial cell, so it is made up of peptidoglycan. So you have to know the structure of peptidoglycan. Peptidoglycan is made up of see, this is the structure of peptidoglycan. Let me explain. This peptidoglycan is having polysaccharides, amino acids. Okay, so what are the polysaccharides present in peptidoglycan? N-acetyl muramic acid and N-acetyl glucosamine. N-acetyl muramic acid and N-acetyl glucosamine. So these two are present in alternate units. Okay, they are linked by beta-1,4 glycosidic linkage. Okay, so several layers of these chains are present. These chains are interlinked by this four structures are tetrapeptide, means 4 amino acids are involved in linking the chains, polysaccharide chains. And again, these tetrapeptides are linked by pentaglycine. Clear? Pentaglycine. So this is the structure of peptidoglycan. So what is the role of lysozyme? Means that will break the beta-1,4 linkage which is present between NAG and NAM. This bond is broken. Clear? So you should not get confused. The bond which is present between NAM and NAG was not broken. Clear? So this is also glycosidic bond. This is also glycosidic bond. And again, NAM, NAG, NAM, NAG, like that alternatively these units are present. The bond which is existing, which is connecting NAG and NAM is cleaved by lysozyme enzyme. So here NAM and NAG, this bond is cleaved. So like that, every NAM and NAG connect, sorry, NAG and NAM. Okay, so again here one NAM will be there. Is it clear? So the bond existing between NAG and NAM will be cleaved by lysozyme. Okay, so this is how it is breaking the cell wall of bacteria and thus it will die. So the exact answer is cell walls. Mitochondrial enzymes, destroying mitochondrial enzymes, no, it is wrong. And lipid bilayers, no, bacteria is having peptidoglycan and we are having lipid bilayers in our cell membrane. The machinery for DNA replication, no, okay. Allergic condition caused by pollen grains of certain flowers causing inflammation of nose is called as rhinitis. Okay, let me explain. Okay, what do you mean by laryngitis? So you know what is larynx. Okay, so larynx easily get infected by different organisms. Due to that inflammation happens. That is called as laryngitis. Okay. And rhinitis is the allergic condition caused by pollen grains. Okay, pharyngitis, again the same, pharynx infected and getting inflamed. Bronchitis, and again bronchioles are infected, so that inflammation of bronchioles happen. Okay. The major phagocytic cells are lymphocytes, mast cells, plasma cells, macrophages. Macrophages is the right answer. Let me explain. So as I told you that the blood is having plasma and formed elements. Okay, plasma and formed elements. So in formed elements, again, you know, formed elements are the cells which are present in the blood. So formed elements, again, RBC, you know the function of RBC, major function is transport of gases, and WBC. So WBC, again, there are different types, granulocytes and agranulocytes. Granulocytes and agranulocytes and platelets. Okay, so if you see granulocytes, eosinophils, basophils, neutrophils are granulocytes. Agranulocytes, monocytes, and lymphocytes. Monocytes and lymphocytes. Clear? Again, there are two different types of lymphocytes, B and T lymphocytes. Clear? So here, these monocytes, again, they are differentiated into mast cells and macrophages. So here, this neutrophils and macrophages are having phagocytic action. So what do you mean by phagocytic action? They involve in phagocytosis. What do you mean by phagocytosis? If you see, this is the macrophage, and if it is facing any organism, okay, so it will go and engulf. It will go near to that organism and engulf that organism inside. Okay, this is called as endocytosis. So by the process of endocytosis, it engulfs the organism. So after the entry of organism inside the macrophages, it will destroy it. So this is the process of phagocytosis, and the cells which are performing this phagocytosis are called as phagocytic cells. Okay, so majorly macrophages and you can say neutrophils are involved in phagocytic function. A person likely to develop tetanus is immunized by administering preformed antibodies, wide spectrum antibiotics, weakened germs, dead germs. So I already explained that. So tetanus is caused by Clostridium tetani. Okay, it is a bacteria that is cured by administering preformed antibodies. So what do you mean by preformed antibodies? So preformed antibodies, they develop in the lab. Okay, so artificially, that is in what can I say? That is in lab, we can produce antibodies against any kind of antigen. If you're having any protein in your hand, you can develop antibodies by using biotechnology. Okay, So for this tetany and snake bite, we are using prepared antibodies. Okay, so this is called as, again, this is called as artificially acquired immunity. Okay, and wide spectrum antibiotics means antibiotics, you know, so many antibiotics, even penicillin, tetracycline, cyclosporin. Okay, all these antibiotics, wide spectrum means they, for example, if you take penicillin, it is wide spectrum antibiotic. So penicillin can kill so many organisms. So if an antibiotic is specific for particular microorganism, we cannot mention that antibiotic as wide spectrum antibiotic. So if an antibiotic is killing so many organisms, we have to name it as wide spectrum antibiotic. Okay, weakened germs, I already told you, if you remove the germs pathogenicity by removing the disease causing part, that is called as weakened germs. And dead germs, so even organisms are, we can heat killed organisms can be used for inducing the immunity as vaccines. Clear? And autoimmune diseases, rheumatoid arthritis, multiple sclerosis, insulin dependent diabetes, all of these. Okay, so here autoimmune disease, already I explained, that is antibodies produced against self antigens. Clear? So the correct answer is rheumatoid arthritis. Multiple sclerosis means that is a virus disease that affects central nervous system. Clear? Insulin dependent diabetes. So what do you mean by diabetes? Increased blood sugar level. Increased blood sugar level. Clear? So why we are having increased blood sugar level means there are two different reasons. Insulin dependent diabetes mellitus, non-insulin dependent diabetes mellitus. So there are two different types of diabetes mellitus. So insulin dependency means maybe the beta cells are not producing insulin properly. So we have to inject those patients with insulin. Non-insulin dependent diabetes mellitus means insulin is produced. Okay. So how this insulin is carrying out its activity? Insulin will be having a receptor on the target cells. So this insulin will go and bind to the receptor to carry out its function. But these receptors are defective. So if the receptors are defective, it won't carry out its function, even though insulin is produced perfectly by our beta cells of pancreas. Okay. Yeah, someone asked about what is the difference between diabetes insipidus and diabetes mellitus. Okay, so let me explain diabetes mellitus. Diabetes mellitus means due to the lack of insulin or due to the defect in receptor of insulin, diabetes mellitus takes place. That is the increase in blood sugar level. Generally, the blood sugar level should be 80 to 120 milligram per deciliter. If it exceeds, we should name it as hyperglycemia or diabetes mellitus. What is diabetes insipidus? Diabetes insipidus means excessive urination happens. Okay, so excessive urination leads to the, what happened, automatically the blood volume will get reduced. Okay, so that leads to change in blood pressure. That is called as diabetes insipidus. Okay. Antigen binding site is an antibody. In an antibody is found between. Antigen binding site in an antibody is found between. So what are antibodies? Actually immunoglobulins. So two light chains, two heavy chains, one heavy chain and one light chain, either between two light chains or between one heavy and one light chain, depending upon the nature of antigen. So you have to know the structure of antibodies. Okay. So there are different types of immunoglobulins like A, D, E, M, okay, G. And there are all the immunoglobulins are having two different types of chains, that they are heavy chain and light chain. So how many heavy chains are present? 2 heavy chains are present and 2 light chains are present. So these 2 are heavy chains. Okay, these 2 are heavy chains. All the chains are linked by disulfide linkages. Okay, so you have to remember the bond which is involved in the joining of all the chains, because in the diagram in your NCERT book it is given. Okay, so here this is called as light chain. 2 light chains, small chains are present and 2 heavy chains are present. Clear? So this is the antigen binding site. This is the antigen binding site of an immunoglobulin. Clear? So the antigen binding site involves 1 light chain and 1 heavy chain. Clear? This is common for all antigens. So 1 heavy and 1 light chain is the correct answer. Is it clear? Okay, the virus that causes acquired immune deficiency syndrome parasites and reduces the cytotoxic T cells, helper T cells, B cells. So helper T cells is the answer. So what is the role of helper T cells and cytotoxic T cells? You should know the different types of cells which are involved in immune system, T cells and B cells. There are no types in B cells, but in T cells, helper T cells and cytotoxic T cells, 2 different cells are there. So what is the major role of helper T cells? This helps B cells to identify the organism or antigen. So these are involved as antigen presenting cells. So they will capture the antigen and present to the B cells, so that they will produce antibodies to remove the antigens from the body. So these are called as T helper cells. So if you are getting infected with the HIV virus, what will happen means that those HIV virus infect the T helper cells and reduce the number of T helper cells. Reduce the number of T helper cells. Clear? So automatically what will happen? The immunity, why it is known as AIDS, acquired immunodeficiency syndrome. So the immune system is getting deficient. Why? It is affecting helper cells, so that even B cells are not completely active. So this is the reason why our immune system is totally arrested by HIV. A certain patient is suspected to be suffering from acquired immunodeficiency syndrome. Which diagnostic technique will you reconfirm? Vidal, ELISA, MRI, ultrasound. So ELISA, let me explain. So what do you mean by Vidal? Vidal is used for identifying typhoid. So here in Vidal test, what will be there? A strip will be there. So there are different types of antigens produced by the organism, Salmonella typhimurium, and there are different antigens. And if I'm infected with Salmonella typhimurium, I will be having antibodies in my blood automatically. Okay, so here in this strip I am having antigen. Okay, O antigen, H antigen, different antigens are there. So if I add my blood, that is infected blood with antibodies, automatically what will happen? Clumping will happen. Precipitation of blood will happen. If my body is not infected with Salmonella typhimurium, we'll be having a clear solution of my blood. Okay, agglutination does not take place. So what do you mean by agglutination? Agglutination means, for example, for blood grouping. So O antigen I have here, O antigen. Okay. So if antibody, if I'm adding for this particular antigen, the cells will get clumped. That is called as agglutination. Okay, and ELISA means enzyme linked immunosorbent assay. So what do you mean by this assay? Actually, a matrix will be there. Okay. Yeah. So I will add a sample here. Automatically, if the antigens are there, I will add the antibodies with. So this is the antibody with the enzyme. So this is the reagent commercially available. So if my blood is having antigen, automatically this antibody will go and bind to the antigen. And here this antibody is already having the enzyme. So I will supply the substrate for this enzyme. Okay. So this product will produce some color. So like that, I will select the enzyme. So an enzyme which convert a substrate to a colored product, I will select and attach to the antibody. This is commercially available. And if the antigen containing blood is added to the matrix, automatically the antibody will bind and enzyme attached to the antibody will convert the substrate to the product. So I will get color. So this is the mechanism in which the enzyme-linked immunosorbent assay helps us to identify the disease AIDS. Clear? Is it clear? So enzyme-linked immunosorbent means enzyme is linked to the antibodies. And if my blood is not having antigen, automatically what will happen? The matrix will be plain. And again, after addition of blood, what I will do means I will slightly wash it off with the solutions. So if there is no antigen, the matrix will be plain. And antibody, after adding antibodies with enzyme, if there is no antigen, antibody won't bind to that. So at the end of the reaction, what will be there? Only matrix will be there. No coloration will be there. Okay. So MRI means magnet resonance imaging. So here, this is used for the detection of cancer. Okay. Ultrasound, again, during pregnancy, this ultrasound means that is the variation in the vibrations, that is sounds produced by the internal organs can be detected, and if it is having any disorders, automatically with the help of ultrasound we can identify. Which one of the following acts as physiological barrier to the entry of microorganisms in human body? So actually what are physiological barriers? I already explained. Clears stomach, gastric juice, and one more thing, saliva. Okay, and even you can say vaginal secretion. So here, yes. So monocytes, they are cellular barriers. Clear? Skin, it is physical barrier. Okay. So next question: out of following diseases which are caused due to the bacterial infection? So typhoid, bacteria. Caused by Salmonella, Typhimurium. Elephantiasis, it is caused by, it is otherwise called as filariasis. That is filariasis, that is caused by Wuchereria bancrofti and Wuchereria malai. Okay, and these are again Vibrio cholerae is the organism causing cholera. Tuberculosis, this is bacteria. Tuberculosis, Clostridium tuberculosis. This is also bacteria. So A, bacterial disease, B, bacterial disease, and D, bacterial disease. Hormone diseases: color blindness, allergic disease, cretinism, and hemophilia. So color blindness, it is X-linked recessive, that is genetic disorder. Clear? So you might have studied in principles of inheritance of variants, genetic disorders. And allergic disease, that is during allergy, mast cells produce histamines. So this histamines and serotonins. So this histamine involves in inflammation. Okay, and cretinism is the hypothyroidism. Okay, so low level of T3 and T4 leads to cretinism. So this is the correct answer, hormone disease. Hemophilia, already we explained, X-linked recessive. Okay. Antigen is usually foreign body which causes formation of antibody. Yes, if you are having antigen, if you are if any infections are there, antigens will be there. That provokes the production of antibodies. This is correct. Okay. Antibody is a protein molecule. Yes, definitely. Antibodies are immunoglobulins. Globulins are proteins. Okay. Antigen can be either of protein and polysaccharide. Yes. What is the biochemical nature of antigen? They are either proteins or polysaccharides. This is also correct. Antibody joins an antigen to destroy the later. Yes, antibody joins an antigen. I already explained the light and heavy chains of immunoglobulin. Okay, immunoglobulin after binding to the antigen, antibody, this will be destroyed. Chemotherapy, radiation therapy, and surgery. So if you are locating, for example, I'm having a tumor in breast, means automatically the doctors will remove by using surgery. So after removal of surgery, the doctors are not confident that they removed all the cancerous cells. So they use radiation therapy. So in radiation therapy, they are using X-rays. Okay, and chemotherapy means they are using chemicals for killing the cancerous cells. So after surgery, chemotherapy or radiation therapy is given, but physiotherapy is odd man out here. This is given for any cramps or any other nervous disorders or muscular disorders. Okay. So the correct answer here is current treatment for cancer does not include physiotherapy. Immune responses are carried out by lymphocytes. Yes, lymphocytes, B lymphocytes and T lymphocytes are involved. Adipocytes are fat storing cells. Okay, they are not involved in immune response. Immunoglobulins, yes, they are involved in immune response. And melanocytes, they are pigment producing cells. Melanocytes are present in the epidermal layer of skin. Melanin production is the major function. They are not involved in immune system. So the cells in the options given here involved in immune responses are lymphocytes and immunoglobulins. So A and C. DNA damage leading to neoplastic transformation, I already explained, that is metastasis. The transformation of normal cells to cancerous cells is called as neoplastic transformation, can be brought about by ionizing radiations like UV radiations, gamma radiations, infrared radiations, and radio waves. So here you have to know what are ionizing radiations and non-ionizing radiations. Gamma rays and X-rays are ionizing radiations. Okay, all other three are non-ionizing radiations. Okay, alteration of which genes lead to cancer? So cancer is caused by the alteration of both A and B, that is tumor suppressor gene. So the name itself indicates that tumor means cancer. Suppressor gene means that is the formation of tumor is suppressed by a particular gene, that is called as tumor suppressor gene. So if this gene is defect, automatically that will lead to cancer. And cell proliferation gene or proto-oncogene. So this oncogene also leads to cancer. The correct answer is both A and B. That's all.